after we're done, it can be downloaded.
Perfect. All right. So I will go ahead and start. We are live. So I want to say welcome to the Soil Speaks podcast, where we give a voice to the foundation of our biosphere, the soil. So in a world where the ground beneath our feet is often overlooked, we will delve into its living, breathing essence, revealing the profound importance of the soil to life on Earth. So our mission is very simple, yet profound: to inspire awareness of the soil's vitality, represented by the ancient symbol of the Ouroboros, undergoing constant cycles of decomposition, regeneration, and renewal, akin to the snake eating its tail. We seek to understand its interconnectedness with all living beings as holobionts, or symbiotic organisms comprised of diverse microbial communities. In this podcast, we explore the intricate relationships between soil, plants, animals, and humans, unraveling the mysteries of our shared experience. So we'll start with a quick round of intros. My name is Everest Holmes. I am the Director of Operations of National Bamboo. We are a company that is committed to creating generational wealth for American landowners using the fastest growing plant on Earth, bamboo. And we see bamboo as a valuable crop that is a new crop with a lot of potential in the United States. However, it has often been perceived as perhaps a nuisance. And so we seek to change that perspective, as well as develop markets for bamboo in the United States. That's what I'm working on. And we take a very heavy emphasis on soil health and growing the bamboo naturally, reducing the need for pesticides, herbicides, and conventional agricultural damaging methods of conventional agriculture. So we really see the bamboo as an opportunity to both create, to serve a need for products, fiber products, food products, and also to, at the same time, let the soil rest, because bamboo is very regenerative to soil. So that's a little bit about me. Max, do you want to say something?
Yeah. So my name is Max Segovia. I've been working in the field of farms, regenerative farms, design, implementation, long-term management positions since 2010. And for some time now, on the side of that, I run a business working with perennial food systems, mainly for people, but I will do any type of design system that they need. I like to focus on the perennial food systems, food forest, grow a food forest is the name of the business. And yeah, I'm a student of microbiology, and I've also attended school for the chemistry side of things, and a few other things there over the years. But that's basically about me.
And we have the honor of having a conversation today with Professor James White of Rutgers University. James?
And okay, thank you so much, Everest and Max. I'm James White, professor at Rutgers University. I've been here 28 years, and 7 years at a previous university. And my work area, research area that students and I are working in is really how plants use microbes, how plants use soil microbiology, how they use microbes in general. So that's basically the area, the general area that we work in.
Excellent. And I understand that, you were very instrumental in the building upon the awareness or the research body regarding rhizophagy, which is a relatively new understanding, correct?
Yeah, it's really, it was discovered, or at least first published, in 2010 by a group in Australia, Queensland, Australia, and they showed that that plants are internalizing microbes in their roots, in their root cells, into their root cells, and we picked up on that really beginning in about 2012. We started looking at that internalization phenomenon into root cells, and what we found is that it's actually a cycle that roots will that all plants will from the tips of their roots, at the very growing root tips, they will secrete exudates, sugars and organic acids, and sometimes amino acids and vitamins even, as attractants to the soil microbes. And soil microbes, they go to those root tips, and then the plant will internalize them into those cells at the root tips. And then they'll extract nutrients from them inside the cells, surviving microbes. They'll actually put in the root hairs and secrete them out of the tips of those root hairs as they grow, so they go back into the soil. So you have this continuous cycle, right, of microbes going in from the soil, carrying nutrients, being extracted from the microbes inside the root cells, and then being ejected back into the soil from the growing root hair tips. Okay, so it's a continuous cycle like that.
That's really incredible. And, recently, after listening to one of your other YouTube interviews and presentations on this, I decided to go and pull out some plants that I started for the spring and look at the root hairs. And I was blown away. I mean, I hadn't at this to date looked at a root under the microscope, looking at it at 400x. And I was able to see some of those features that you're describing. Max, have you had a chance to check out roots directly?
Yeah, not the roots themselves, but I have extracted from the roots into dilution and looked at it and seen all kinds of stuff. Yeah.
Well, that's a fascinating topic in and of
The
itself.
roots, leaves, yeah. I've done leaves too, a whole bunch. Yeah.
Mm-hmm. Mm-hmm. Yeah. So this, James, what you're describing, this rhizophagy cycle is just happening in the tip of the root. Those microbes are not there. Are they traveling further beyond the root system?
They do, but the rhizophagy cycle stays in the roots and goes around, right? But some of those microbes that are in the, and by the way, that is happening right at the root tips. It's happening where the root hairs are. So older parts of roots, you don't see this happening. So just right at that root tips, which is where you do see the root hairs, that's where the cycle is happening. And those root hairs will eject microbes from their tips, so long as there's microbes in there. They'll grow and they'll eject microbes so long as the microbes are there. If there's no microbes, you get no root hair growth. Okay, so really, you have to have these microbes in the root hair, in the root cells, in order to get root hairs to grow at all. And it is to the point that you made, Everest, that this can easily be seen, visualized by people. And we actually I've done a couple of demonstrations, public demonstrations, and both of them actually with some guys from Mass NOFA, right? The Northeast Organic Organic Farming Association, at big shows to actually demonstrate these bacteria coming out of the root hairs and in the root hairs, right? Actually, I noticed, I watched a YouTube presentation by Matt Powers on this, and he showed also in some of his demonstrations the bacteria in these root hairs. So it's easy to see them. You just have to kind of look for them. It's surprising people haven't seen it before. I mean, it really is surprising because it's so easy to see them. I think, the barrier? The
Uh-huh.
barrier is not outside us. The barrier is in here. That's the barrier because
Selective processing or
see what
awareness.
you see awareness, right? If you think that eukaryotic cells or plant cells or animal cells don't have microbes in them unless they're diseased, right? If it's a disease agent, then if you see something in there, you don't think it's a bacterium. You think it's a vesicle, and that's what people have reported them as. They've
Hmm.
they've I look at some of the literature, some of the old literature, and they'll have pictures of root hairs, root hair tips, the root hairs themselves. And we
Yep.
can clearly see the bacteria both inside that root hair in their picture and coming out, accumulating at the tips. And they never saw it. They never saw it. In fact, they called them extra, what was it? Extra cytoplasmic vesicles. That's what they call it. They have to make up a special term to describe these bacteria coming out of root hair tips.
Wow.
But
Wow,
it's in
that's
the
amazing.
brain. It's in the brain. That's the barrier. It's not out there. People could figure it out if they thought. Everybody could figure it out if they thought that they're in there. Then they could see them real easily. But thinking that they're not there, they don't see them. It's invisible. They're basically invisible to people.
Yeah. Yeah. And agriculture as an industry has become so reductionistic that
Right.
perhaps it's overlooked simply because there's a system that seems to work, right? That's established. And do you think that there is a way to harness this cycle, this rhizophagy cycle for agriculture that's straightforward?
Well, I think there is a way to use it. I mean, just knowing that it's happening
Mhm.
is the first step, right? I mean, it's like you got to admit you got a problem, right? But you got to work on that. If you're going to solve a problem, you got to admit you got it first.
Yeah.
So in this case, if we understand the process is going on, if we, and this is important for growers and gardeners as much as it is for people trying to produce products for growers and gardeners and homeowners and so forth. If they realize it's happening, then one could then, for example, be careful of how we treat our soils and build our soil microbiology, build organic material in the soil, use compost and so forth, use cover crops, and different cover crops will actually contribute different microbes to the soil that the plants can then get. And kind of understanding that rhizophagy cycle happens is the first step, and then starting to build up the microbial populations that work with the plants. That's the next thing. Further on beyond that, one could, and we've worked with some companies already to help them evaluate whether their microbes are working in the rhizophagy cycle. So that's the next level for companies, right? To kind of select their microbiology so it's optimal in that system.
Mm-hmm.
So that's the yeah. So I think that it
That's
is.
the golden goose in a
I think it's possible.
country
Well,
company.
yeah, but nothing is magic. None of this is magic.
Right.
It's all work, and plants have to, plants use more than one microbe, really. So
Absolutely.
yeah.
Yeah, I think that's definitely a direction I would like to go in this conversation at some point. I do want to zoom back out for a second and ask you, James, like, what inspired you to really delve into the rhizophagy cycle? You mentioned the research in Australia, but was it?
That did not. Yeah, that research wasn't the beginning
Mm-hmm.
of the quest that we were on, right? I mean, this quest actually started back when I was an undergraduate student
Oh, wow.
in Alabama, back when I was, it must have been 20, 20, working in a lab there with working on a fungal endophytes in grasses. And that was where it started. And I
Okay.
was just a work study student, Yeah. And so I was working, my father was military, we didn't have a lot of money, so I got work study and financial aid and so forth. So I was working in that lab and learned how to work on these fungi inside these grasses. And then I moved on, worked on my PhD, studying again these endophytes in grasses, and did a lot of work then. And then about it was a few years ago, 12 years ago or so, we started looking at bacteria. It had been fungi pretty much up to then, and using the same kind of method that we used, which is a microscopic method that we developed for working on fungal endophytes. So we used similar methods, but we had to modify them because the stains didn't work right, because these bacteria didn't stain like fungi. So we had to develop some new stains. And so one thing that we did is we developed some reactive oxygen stains. One in particular that gave us hydrogen peroxide staining. And when we used that stain on plants, particularly on plant roots, it was amazing. Plants, we could see everything inside, all the microbes that were inside those root cells and the plant cells. And the reason for that is because there is a hidden process going on inside plants that nobody realized was there.
Right.
And that is this internalization of the bacteria and then the manipulation by the plant of the bacteria using reactive oxygen, like superoxide. Superoxide is this potent form of oxygen that reacts in a big way with biological molecules, and particularly these bacteria. And so the plants were secreting superoxide onto these bacteria. And of course, the microbes are doing something inside. They're interacting in their own way. And I'm not going to go into those details, but there's this interaction. The point is, when we looked at this reactive oxygen stain, using this reactive oxygen stain, and we looked at the roots, you could see they were filled with microbes.
Wow.
And that's really how we started studying the process to figure that out, exactly what's going on. And then it just took time with the students and they're actually my lab manager is a lady named Kate Kingsley. She and I were working very closely together at the microscope, working on some of this material, some of this plant material, looking at this. And sitting there together, we figured out that the bacteria are going in at the root tips and being ejected from the root hair tips to make that cycle. And that's when we knew that it was a cycle, And when, from the time we saw the first microbes inside the root cells, through that whole exploration of understanding that they were cycling through plants, before publication, this happened probably 1 or 2 years before we published anything. We were studying it, trying to understand, looking at plant after plant, and during that time, it felt like we had this big secret that was going to be earth shaking. That was going to be earth shaking, and it just felt. There was just so much satisfaction. I actually didn't want to publish it because it felt so good to just know how important this process was, But we finally did publish it in 2018, the first publication. But we'd mentioned it in a couple of articles before. 2017, we mentioned it, a couple of articles. We talked about what was going on, but we didn't really define it completely. But in 2018, we published it, defined it completely, and then after that, there was basically the phase in which we talk. I go on a lot of talks and explain it to everybody, and we worked out the further details and over the next couple of years. Still working on those details, but basically we published everything, and we talk about it now. There's this phase of the work.
Everything is published now.
Pretty much everything is published now, and we're talking about it because I think, and the reason we're talking about it, because I think it's important information for all of us to know that
Absolutely.
that plants work.
Priceless, yeah.
That plants do this. I mean, this is something that a lot of people thought already. They thought this already, that something is going on. They didn't realize, didn't have a name for it. They didn't know there was a cycle involved, but they thought that microbes in soils are critically important for plants. Okay, and the rhizophagy cycle is how that is manifest, how people can see that.
Right, because the way I imagine it is that these bacteria are literally the food for the nutrient. They're supplying directly. The plant is feeding on them, right?
They are, and they're managing them. They're managing the microbes in the soil.
Yeah.
They're absorbing them into their cells, and they're managing them in the cells. They're using that superoxide. Once they absorb those microbes, those bacteria in, they hit them with superoxide.
Mhm.
That superoxide oxidizes the cell walls off of the bacteria inside the root cells, and then takes the nutrients that were on those cell walls. And we think cell walls, bacterial cell walls, carry, in the soil, floating around in the soil, will pick up metals and things like that, nutrients on their walls and in their walls. And when they get absorbed and they lose their cell walls, the plant gets all those nutrients that were on those bacteria. And not only that, once the walls are removed, then the plant will replicate those bacteria. It will move them in such a way in the hairs that causes them to break up into little, I call them clones. But essentially what it's doing is makes them divide. It makes them divide, and they divide very fast without cell walls.
Oh, wow.
Because the cell walls is what really takes time for cell division.
That's
So they're dividing a lot, so they get a lot of copies, and then those microbes will, in producing hormones, ethylene and nitric oxide, will cause root hairs to elongate. And as those root hairs elongate, bacteria are ejected. They're protoplasts without their cell walls. So those bacterial protoplasts go out of the tips of those root hairs. And there's little breaks, because that's the growing part of the hair right at the tip. And that's where the breaks, the little pores happen, and that's where the microbes are ejected out. You can see them being ejected out. You can see them. The other thing that's happening inside the root hairs is they're being moved around like that inside the hair. And that almost nobody sees. You have to look, because what happens is that's a cytoskeleton, a cell skeleton activity. If it has an attack cell skeleton, which is actually protein, the root hair can move the microbes around like that. That causes them to break up. It breaks them into bits, makes copies of them, right? Makes a lot of
there's
copies.
a circulation inside of each individual cell of the root, and there's a cycle where the bacteria are being ejected out of the root hairs.
Okay, let's admit it. It's cycles within cycles, right?
That is
Cycles
incredible.
within cycles of nature. Cycles
Wow.
within cycles of nature.
Yeah.
Wow.
It is. It's really cool. But the point that I was trying to make,
Yeah.
That's a great point that you made, Everest. I like that. The point I was trying to make is, if you pull the hair out, pull the root out, the hairs turn like that, and that breaks the skeleton. And then you look at it, you can't see it. So what you have to do is you have to grow the plant in a medium that's clear.
Oh,
And the root
that was
goes
my
down.
next question. Yeah.
Yeah,
So
and
this
then
is
you
how
turn
you-
that upside down, right? And say it's an agarose or agar or something like that. We use plastic Petri dishes. We should use glass ones, but they're too thick. But anyway, we use plastic Petri dishes, and we turn them over, and then you can focus through the bottom of the Petri dish, and you can see the motion.
So as a citizen scientist, microscopy is using microscopy to observe this. If Max and I wanted to do that, we would need to germinate seeds on an agar so that we could actually observe the ejection of the bacteria. That's what you're saying?
I am suggesting that, but you could, you don't have to do that.
Okay.
Because you could see immediately post ejection. And I have observed ejections very soon after moving, but they're probably triggered injection. Pull them out and you trigger them because of the, water rushes in, and that causes an ejection, right? So you can see microbes ejecting. But if you want to look at it over time, then you have to do exactly what I said, and that is grow it that way, turn it over, look at it. But you can see if you pull them, what you can see are things like this. You could see fat root hairs full of bacteria. You could see that. And you can see bacteria emerging from the tips, clusters of bacteria at the tips. That's the kind of thing you can see. You can also see, sometimes you can see transitional forms where bacteria go from inside to out, and you see, and that's actually the test of, that's what we use as a continuity test. It's something I learned when I worked on fossils many, many years ago. With fossils, they're frequently disarticulated into pieces, right? Lots of different pieces. You don't know what connects together. So to put together an organism, or to put together the cycle of the organism or the structure of the organism, you have to find where those parts all connect. Okay, so that's how we do that. Also looking for those transitions where you see bacteria in, some bacteria going out, and then bacteria all outside. You look for those transitions. So that's just a way of doing microscopy work to kind of tell how things related to one another.
I see.
Now, what type of microscope are you talking about? Like electron microscope? I mean, we use shadow microscopes for what we're doing.
Just a light microscope.
Yeah.
You can use just
Right.
basic. I use a fancy light microscope, right?
Yeah.
But it's pretty old. I mean, this is a microscope. When I came to Rutgers, I think I paid 25 grand for it. Then a Zeiss, nice Zeiss microscope, compound microscope with, all the bells and whistles at the time. You can get them now for 50 or 60,000 still. Really good microscopes, but you don't have to have that fancy microscope. You could use a much less sophisticated microscope. They're still pretty good. You go to high dry, and you could see fat root hairs, and you could see the microbes around the tips of the root hairs. You can do fluorescent mode. That's what Matt Powers did. Is I think he was using fluorescent microscopy or something like that. And we just used the DAB, the diaminobenzidine the hydrogen peroxide stain, just to on the public demonstrations that we did to show rhizophages cycle. And the reason we did those demonstrations is probably not so important now, but I wanted people to see really how easy it is to see this going on. So it's not like science. A lot of our science, it's cryptic. It's hard for people to see, but this is not hard for people to see. Once they see it, they have, they're empowered.
So we've described the bacteria cycling throughout the root hair back into the root tip. And part of that, what you're saying, is that it's a nutrient acquisition for the plant. So the bacteria, maybe, correct me if I'm wrong, but you're saying the bacteria are being degraded, the inside of the cell, the plant is taking those nutrients, the bacteria are dividing, the protoplasts are remaining intact as a living bacterial cell. But then when they exit the root hair, they are able to acquire more of nutrients to complete their cell body again, and then they go back into the cell. Basically, that's how this is bringing nutrients in.
Yeah, I'll explain a little bit more. Okay, they take these microbes in, all kinds of microbes from the soil, right there. There's communities. Now, not all microbes go in. Most of the soil microbes don't go in, but some of them do. And those are the ones the plant takes in. Some of those microbes get totally broken down. If they have no resistance to the superoxide, they're just degraded. They're just fully degraded. And we can see that inside. We could see that in the images that we make. We could see them losing proteins. We could see bacteria swelling and disappearing. But we also can see bacteria replicating in there. So there's this replication that continues to go on. It's like a race inside the cell, inside the plant cell to survive. Because if they don't constantly replicate, the superoxide is going to oxidize them away completely and they won't survive. So there's this race inside the plant cell. The survivors of that race end up in the root hairs, and they will accumulate at the tip of the hair. And then at the tip of the hair, they'll secrete hormone. And the hormone, well, first it's actually at the root hair initial, which is just like a little bump on a root cell, right? On the root epidermis and the root skin, little
Right.
bump. And so the bacteria go in there, and in mass, they secrete hormones, growth hormones. They secrete ethylene and they secrete nitric oxide or other nitrogen forms from what we can tell. And then those trigger elongation. Those are known to cause root hair elongation. And they do, because when we remove those microbes, or we block the formation of ethylene and the formation of nitric oxide, we don't get those root hairs forming. So we can either by removal of the microbes or blocking the production of those substances by the microbes, then the root hair doesn't elongate. So we know that's happening. But when that root hair elongates, it will actually cause the bacteria that have accumulated in the tip of that hair. It will cause those protoplasts to go out of the little pores at the tip of the hair. And when they go out, the root hair puts a little bit of exudate out with them, some exudate, right? Some nutrients that go out with them. And those exudates allow those bacteria to reform their cell walls. And we know that because what we see there is we can see when they first come out, they don't have their cell walls, but then and they're also tiny. And then a little bit later, a little bit of distance from the hair tip, we see they begin to get their cell walls back. So you see that transition beginning inside. They're just these little protoplasts, tiny little protoplasts. Outside, they begin to get a little bit bigger. And then a few older, I don't know how many, probably hours older. I don't know how much older they are, but you see bigger microbe cells with their cell walls fully intact. So, and it's known that root hairs put out exudates from the tips of the hairs too. That's been previously published. Yeah, so that, I think, was there another part of that question? I feel like I'm missing part of that
Well,
question.
I guess I was trying to understand if when the bacteria, so what you just described really makes sense, and that's why you make the statement, the profound statement that plants are farming microbes, right? But then my question was, are the bacteria, once they exit the cell, are they mining raw mineral nutrient? And then
they do that automatically. Yeah,
Yeah.
they do that.
Yeah.
They mine. We call it microbial mining. You've heard of that before too. Yeah, it's someone else came up with that idea, but it fits quite well. Yeah, that's what we envision is happening. They're mining, and the plant will also help with that, because the organic acids, for example, that plants are putting out are solubilizing. They're solubilizing phosphate, stuff like that. And so that solubilized phosphate and those other nutrients that get solubilized, the microbes just pick them up. They eat them up. And it's they're the perfect vehicle for carrying back nutrients to plants. The other thing that's happening is that some of these microbes are nitrogen fixers. So they take these nitrogen fixers in, and they'll use them in the root hairs, right? There's a process that happens in the root hairs where nitrogen is involved, and so they'll get some nitrogen from that. They'll move those microbes up through the plant, up into the leaves, into the trichomes, and into specialized cells in the plant that typically are not doing photosynthesis, or there's some protection from the oxygen, because oxygen can actually reduce nitrogen fixation by these microbes. So there's certain tissues and cells where more nitrogen fixation can happen inside the cells. And so the plant is also doing that and getting nitrogen. In fact, I want to add another part that it is about nutrients, and we measure like the divalent metals, really important for rhizophagy cycle, divalent metals, the zinc and the manganese and the magnesium and those important nutrients for plant. Those are really important for rhizophagy cycle to deliver. That's really important. But the other thing that is, and all nutrients are important. So, but the other thing that is significant, maybe even more significant than the nutrient issue, and that is that plants do not develop properly without microbes in their tissues. They can't grow right. They have to, the microbes are part of the plant system for growth. And these microbes go all over plants, and they are producing hormones and nitrogen, and that's causing cell growth in plants, but also having some other effects. And that is they're affecting DNA of the plant. They are both ethylene and nitrogen, nitrate in particular, are known to be what are they called? Nucleomodulants. Nucleomodulants. They will affect the DNA of the plant. And what they do, it's really interesting. What they do is they cause DNA replication. They cause DNA replication. You can find the literature out there. And this is called endoreduplication, or also endonucleation. I think there's different terms, or just reduplication.
And the reason for that process would be in the plant would be
well, they're interacting with the plant, and that's part of what causes cell growth, is that they cause the DNA of the plant cell to replicate, and then the plant cell gets more cytoplasm because of that DNA replication. But the outcome of that DNA replication is that they're affecting in a big way
stress tolerance and
epigenetics.
The genetics of the plant.
The plant are being impacted, and so you get these highly microbial plants that are filled with microbes that actually look highly variable, because they develop various ways. You can see little ones and big ones, big leaves, so it affects development, right? These microbes,
their capacity to adapt.
Yes.
Precisely, Max. Precisely. And so when we have climate change and we have perturbations that are coming, what we really need to do is to start working with these endophytes to produce plants that can adapt faster. And it's more than just adapting in terms of growth and how they look, but the fact that the microbes are in there and they're interacting with the plant via reactive oxygen, right? Reactive oxygen is in there. That makes plants become more resistant to oxidative stress, to any kind of reactive oxygen. And that's oxidative stress. All stress is oxidative stress. Salt stress is oxidative stress. Heat stress, it's oxidative stress. So when plants produce antioxidants and phenolics and things, that because they're interacting with these endophytes, endophyte, by the way, this is just any microbe that goes in a plant and does not cause disease. Internal phyte, plant, internal plant, right? Endophyte, these microbes that go in. Because the plant is interacting with them with reactive oxygen, these plants become stress hardy. So they can tolerate heat, they can tolerate salt, they can tolerate a lot of stresses that our conventional cultivars cannot.
Yeah. On a side note, that's one of the things that attracted me originally to bamboo. Back when I was in college working on the garden crew, there was a bamboo grove there. We were producing food for the college cafeteria, and then this bamboo was there, and I got really interested and started looking into it. You know, among other crops, ginseng, other potential high value crops that could be more widely adopted and grown in the United States to help farmers. And bamboo struck me as this really important overlooked crop because and it turns out it's such a primitive plant grass that it has many endophytes compared to conventional crops, monoculture crops like corn. And so it does, and bamboo's known for its kind of resilience. And, people will often make a kind of a little bit of a greenwashing claim that bamboo doesn't need pesticide or herbicide or fertilizer. But really, that's not the function of the bamboo as much as it's the function of the endophytes and the microbes. Because if you strip them in, for example, tissue culture, you strip the bamboo tissue of its endophytes, then you have a negative outcome, growth outcome. But the bamboo is this primitive plant with such an amazing endophyte profile that it seems to endow it with these qualities that it's so well known for, the stress tolerance, the pest resistance, and so on. And so yeah, it's fascinating to learn more about this in regards to that.
So bamboo
Yeah.
is an amazing plant, and the rate that they grow, the amount of carbon that they can produce, the organic material they produce, is undoubtedly, and I have never had the chance. I mean, I've looked at for fungal endophytes years ago in bamboo, but recently in the past few years that we've been focusing on bacterial endophytes, I haven't had the opportunity to look at them. But undoubtedly, there are some secrets in bamboo that are yet to be emphasized. And I'll give you an example of something that I would like to look at like to be able to find in bamboo, and that's just, or something comparable. But as we're looking at this invasive Japanese stilt grass,
Okay.
this is a basic plant, right?
Yeah,
and,
I
and
think
it
I know
moves
exactly
in.
what you're talking about.
You know what I'm talking about. It moves in. It's moving in little by little. It's in the United States now, and it's moving into our cultivated areas now, cultivated little by little. But real Japanese stilt grass. We have a weed scientist here, Matt Elmore, and Matt Elmore and I have a graduate student working on this, Carmeline Rodrigo Pule, her name. But what she's looking at are these endosymbiotic interactions, nitrogen fixing endosymbiotic interactions inside this Japanese stilt grass. And what she is seeing is that they're inside these leaves of this Japanese stilt grass, there are files of cells, rows of cells, about 2 or 3 rows thick, that stripe through the length of the root. Wow. I mean, the leaf, not the root, the leaf blade, and stripe through it. And so you can see you have a layer in the root of the leaf that has 2 or 3 cells thick that are full of bacteria that are fixing nitrogen without chloroplasts in there. And then you have mesophyll that are, several layers thick of mesophyll, 4 or 5 layers that are where photosynthesis is being produced. So and then you got another layer where nitrogen is being fixed of these, bacterial containing cells, and then like that. So this leaf is striped. Once you stain it, you have to stain it now for nitrogen. But once you stain it, you can see these specialized cells in there. So
it grows so good. We have it all over our property, man. It's just, you can't stop it almost.
Well, I'm thinking we'll find something like that in bamboo.
Yeah.
I'm thinking bamboo.
I have a quick question as far as it seems like a lot of these studies are being done mainly on plants and such. And one of my huge interests is how much of this is being investigated in trees, and how difficult is it to investigate it, particularly in an adult tree. So
It's a great question.
It is, we're trying to work on that. Not much of it. Here's the answer. Not much, but we do have a Lena Struve, one of the a plant evolutionary biologist, taxonomist here, right? She and I have a student working on tree endophytes and trying to figure that out. And Maxine Marbosa is her name. She's a forest person, but a PhD student candidate. But we looked recently at a crabapple, and we saw that in the recently formed, and this was just a couple of weeks ago, leaves, we looked at some of the mesophyll cells, and those mesophyll cells actually contain bacteria just underneath the cell walls. So we're looking closely at that, and looking at some other trees as well, trying to find these. But to find this stuff, it's kind of hard to say, okay, I'm going to look at this plant and go and study that plant and find it. Instead, the most convenient way is to say, okay, I'm going to look at a bunch of plants and I'm going to see what pops up, kind of from what would you call that? Just count on some luck, right? Count on some serendipity. So start looking at and see where you see it. And then when you can see it really good because the tissue is right and the plant growth stage is right, and you can open it up and see it well, it doesn't get it. Okay, so doing that, you find a plant where you can see it really well, and then you study that first. That's the first one you work on, right? And then the ones that are harder, well, okay, we'll save those for later, because the first thing that has to happen is you have to make the point, right? And the point is that we're trying to make is that plants have developed ways, specialized cells and tissues in which they will juice microbes for nutrients. That's the point. That's the point. So that's a big, once people understand that, then you can go in and look at all the details, right? But the big principles first. So that takes a little bit of luck to find the systems where you can make that kind of progress doing that.
So that really brings up a huge point, which is that, conventional agriculture is very reliant on synthetic fertilizer. And I mean, not even necessarily synthetic, but on fertilizer. So in your opinion, and maybe the answer isn't so well known yet, but obviously there's these different sources of nitrogen, different mechanisms, and then there's a lot of biodiversity with the biology, the microbiology. But you see what I'm getting at? What is the total, maybe you could say, estimate of nutrient acquisition by plants utilizing endophytes or symbiotic mutualistic microbes in the soil?
Well, if you go, if you go look in one of our cultivated fields, I think you'll find them, especially if there's not, they haven't cultivated microbiology, they don't have good, and they're putting fertilizer down, right? They're putting, in this case, let's say it's chemical fertilizer, right? Lots of nitrogen, lots of phosphorus, lots of potassium. Yeah. If you look at those fields, then what you'll find is there's a minimal amount coming from microbiology. It's all pretty much coming from the chemistry that we're putting on, right? Now those plants have microbes. Well, that's what's happening, and we're getting all that waste in the environment, right? All the environment, which is a big problem. The damage, and the plants are not well either. The plants are sick. That's not how plants work. Okay, but if you go to nature, you go out to nature, you go in the woods, and you find a plant. Very little of that is actually solubilized chemistry, right? I mean, yeah, some of it is, not that much. Most of it is microbiologically determined. I mean, even if we consider microbes around roots that are solubilizing, right? That's microbial source. That's a microbial source right around the root. that would solubilize some, but mostly you're using mycorrhizae, they're using rhizophagy cycle, they're using bacteria, microbes inside their tissue,
Yeah.
get them. They're doing all of that. That's how plants work in nature.
Yeah.
In agricultural fields, in using our chemical agriculture paradigm, which is what we mostly have, and it works, you
Mhm.
can put it out there and grow plants, and they can grow pretty big, and yeah, you can grow plants fast using all that chemistry. But of course, that isn't how plants actually grow
Right.
in nature, and they become, what happens is the plants are weaker. They're not, because they're not doing oxidative interaction inside, so they don't develop that resistance to reactive oxygen, to stress, right? They're not as stress tolerant. The other thing is you get a lot of disease on plants, and you don't get that in nature, because the microbes themselves will actually, besides making plants more resistant, they'll colonize the potential pathogens, and they cause pathogens, fungal pathogens, for example, Fusarium or Alternaria, or any fungi out there that have both a saprophytic mode of existence and a pathogenic mode of existence, right? What they do is push they push the facultative pathogens, right? They push those microbes, those fungi, into their saprophytic phase, and they become more avirulent, less virulent. They don't cause disease. It is incredible. It's a whole system. This is what
It
actually
is.
And you are, James, if I understand correctly, you are a degreed plant pathologist. So you're speaking with authority here when you say that.
I am. I'm a mycologist as well, a botanist, and we wear a lot of hats in academia.
Yeah.
So one of them is as a mycologist, and we see, even in mushrooms, we see mushrooms doing very similar things, taking bacteria into their hyphae and into their fruiting bodies, and using that to get nitrogen. So it's more common that people realize. The microbiology is critical for plants and critical for the whole system. The pathogens that affect plants, it's critical. It's critical to have that there. We'll have a lot fewer. I'm not saying we're going to get rid of agrochemistry completely, because those are tools that we all developed, and people need that. If there's a crisis, we have to have something so that people don't starve to death, right? But we don't need that stuff, and it's causing, at least not unless it's an emergency, and it's causing us to use more and more chemistry.
Wow. So if you were if there's a farmer out there listening to this, and they are, interested in obviously becoming more biological rather than chemical, and but they've got a serious fungal pathogen or pest, or like take for example the citrus industry as a great one down in Florida, dealing with greening. What are you saying that the solution is not it's never going to help you in the end to use fungicides? That's just going to perpetuate the issue. It's the solution then is what?
I absolutely am ever saying that the solution is not kill microbes. The solution is add microbes. That's the solution, which seems counterintuitive, but that's what we do. And we think, in fact, in the laboratory, based on experiments that we've used, that we could grow plants entirely with microbes. We don't need any chemistry, no fungicides.
Oh wow.
Even though herbicides.
I don't use any chemistry in the systems that I manage, and I've managed them for many years. And I'm talking about not just food systems, but straight up landscaping systems that are fully managed biologically, which I don't really hear of anybody doing around here.
I believe you, Max. I believe you, because that's what happens in Mother Nature. That's what Mother Nature does.
Yeah. And you were just mentioning, and I know that Everest and both of us in school, we've seen anybody can go out there and quickly you do a quick search and you can see the data of the available plant nutrients that they find in an old growth forest in comparison to agricultural field, and how low the percentage is in the old growth forest,
Right.
for the simple fact that the microbes are just slowly cycling nutrients, and the whole process is slowly, little by little cycling nutrients as is needed, as opposed to us dumping all this nutrient on there. And
Right.
then, so yeah,
Yeah, to
it's
your
very
point.
obvious.
I was just going to say, to your point, Max, that's not necessarily a good way of measuring the productivity, right?
Right.
Yeah.
Yeah, I
Um,
think we've just been, you know, we've all been accustomed to the idea, or we bought in, all of us bought into the idea that we have to use chemistry to grow plants. But
mhm.
when you analyze it, as soon as you analyze it, you start thinking about what happens in nature, or what people have done, like what Max is doing,
Mhm.
growing plants without adding any of that. And then you start to question that. And I think that's really the first step to all of us, adjusting how we grow our food crops, grow crops. So it's just realizing, just that realization that that's not really how plants work. They don't
Well,
really chemistry.
it, it,
Go
it's
ahead.
funny to think that billions of years of evolution are going to be wrong over humans coming along and saying, no, this is the right way to do it. Bottom line, that's
Right.
always the thought process for me. When it
Yeah.
comes down to it, there's a reason for why it was perfected by nature for a long, long, long time, for us to come along and say, oh no,
Yeah, and that
chemistry is the way
also
to do it.
in this conversation has brought up for me a lot of emphasis in the current paradigm is on, genetic modification. And James, based on what you're saying, it seems as though that a better focus would be on not genetic modification, but let the microbes kind of do it. Like we should be focused on microbial diversity. Is that essentially it? Microbial diversity is the silver bullet in a way.
Well, I think it's it's simpler, really. Um, I personally think that the solution is focusing on soil health,
Mhm.
building up soils so that we have the organic material in there, and it's composed of the right stuff, right? And then focusing on the microbiology, growing the microbiology, and that's going to, basically regenerative agriculture, tenets of regenerative agriculture, right? I think that's the solution.
Mhm.
The other part is endophytes in plants, and that is these plants go with the seed. The mother plant will put on the seed the microbes that work best with that seedling. So taking care of those microbes that are on the seed, right? Not putting coatings on, not sterilizing seeds, stuff like that, right? Taking care of seeds so that and then if we understand, what is a healthy plant, how healthy plants grow, you can tell a seedling, if it's a, if it's a weak seedling, because the microbes have been killed, it's been grown with fertilizer too long, and there's no microbes there. You could tell when you start to grow it without all the chemistry, they don't do well. You know,
Mhm.
so, so that's the so basically selecting the right seeds, right, and taking care of those microbes, and then building up soils. I think that really is the key there. Regenerative agriculture, right, and the other part. I don't have any real issues with genetic manipulation. People are going to do that. There's no way to
Sure.
stop it, really. People are going to do it. This is what humans do. We
Yeah,
play with stuff. We're going to be doing that. Well, of course, we have to be careful with that.
makes sense.
It would be better if we understood what we're doing. And that's the main thing that I put out there. I say, our knowledge is fragmentary, and just turning off pathways and stuff like that, truncating pathways or whatever is done. There's a reason those pathways were there in the first place. And what are we really doing? We're probably not going to make a monster by doing stuff like that. We're going to make microbes that can't survive. That's what we're going to do.
Mm.
But
I'm
never,
sure
go
you
ahead.
have heard of the idea that there's, more microbes in a teaspoon of soil than people on Earth. And and we only have 40 years, 50 years left of topsoil. And, I can even look out into the forest and see that there's issues with the forest health. And I would hypothesize that's due to the fact of deforestation 60, 70, 100 years ago, and we just lost that soil. But my question, I guess, is that, you know, is soil health an existential threat to you? Do you think soil health is an existential threat, or are we being dramatic?
I don't know that I would say it's an existential threat, but I would say that uh, the solution to a lot of what plagues us now in society and on our earth, soil health is the answer. And that is that if we take care of our soils, we start to grow plants differently, we're not going to have all the contamination. We're not going to have the all the nitrogen going into the oceans and the waterways. We're not going to have we have to use so many poisons that are poison people, then we try to use it, right? They're not going to have those toxic residues on our foods. We're once we move down this other path, the biological agriculture path, it's going to be we're going to have a cleaner world. Our human health is going to be better. Our own microbiomes, don't come from us eating pills and stuff like that, or
Exactly.
comes from the microbes. It comes from the microbes that are on the plants that we eat. That's where our microbiomes come from. And you can tell that because if you look at people who live in the forest, the pygmies and people like that, they have used tremendous microbiomes. They're highly diverse gut microbiomes. People who go there and stay with the pygmies for a little while develop their microbiomes. But again, when they leave again, it goes down to our modern.
That exact thing has happened to me.
Yeah, did it.
And spending time in the Amazon jungle for 3 months, my stomach flora became practically perfect. I was drinking from the river every day. I couldn't believe my eyes. Any of my skin issues, any of the things that were going on up here up north, they all went away. And I kid you not, I came back home. I've told others this story. I came back home, and the very next day, I woke up with an allergic reaction.
Wow.
Rash, and I was never the same again.
Wow.
Go ahead, Max. I'm sorry I interrupted.
Yeah, no, that's it. I mean, for years I pondered with it. I started my journey out as a student of medicine, really, and before I switched to plants. So that whole thing, how it ties to the human microbiome and the human health, is, and both Everest and I are hugely interested in that. But Absolutely. For years I pondered on what it was. What part of my diet was it? And then it wasn't until I got more into the study of microbes that I realized that honestly, I think that the biggest difference was drinking from the river every day. The amount of microbiome that I must have been taking in my body from drinking from this clean, pristine, up in high in the mountains, in the tropical region, it must have it for sure changed temporarily my microbiome.
Related to that, we are creative. We have people who are so creative. We develop all kinds of science, right? We have engineers, food science engineers, who know how to do things, basically keep microbes out of food. And my daughter's an engineer, so I don't have anything against engineers. It's just that we get these ideas, that food has to be sterile, and we do it, and then everyone is eating that. Now sterilized foods, and so it's it's
Yeah.
counter what's good for us. That's not good for us.
Right. It's my kind of vision of the future that, and I'm sure you all might share this, but when I think of a future where we have more healthy food choices, it seems as though the clear path forward and the most rapid path forward is that people, the society, reclaim a little bit of agency in their own food production, right?
I agree. I think that's right. I think that we should everybody we should all have a little garden space somewhere and grow a little bit, even if it's just a few little tiny grape tomatoes or something like that, grow a little bit to kind of connect with that. And there is a lot of people are saying this, and there is a connection between soil health, plant health, and human health or animal health.
Mm.
And that connection is microbes. That connection is microbes. Of course, it's also nutrients, because we know that plants produced with microbiology are in general at least 30% more nutrient dense, right? Nutrient phenolics and micronutrients, metals and zinc and iron and manganese and magnesium and stuff like that, right? Those nutrients, they're 30% or so more than conventionally grown plants. And that is the microbial contribution. That's what the microbes are doing. And of course, you're getting those microbes in addition to those nutrients. It makes a difference.
What would you say to people, maybe engineers or agricultural researchers who are so focused on the solution being aeroponics or hydroponics, indoor systems? There's a lot of I just see in the space there's a lot of emphasis on that, and I don't buy into it personally. I'm sure Max, you're advocate of food forests, and I'd love to hear you say something about that too. But what do you think, guys? Like, what do you say to somebody that thinks the way is, automated indoor sterile systems?
Okay, Max, okay, if I answer first?
Please.
Okay. Well, of course, hydroponic and aeroponic systems, they do work. They work, you can grow plants, but the issue is you're essentially feeding them everything, and microbes work too. You could give them biostimulant microbes and we can see a difference. I have a student working on that. You can see a difference, but of course, you're guessing what the plant needs and you're putting it in. Plants don't really need the microbes. And so they're not really going to be developing this. So you have big problems with diseases in these systems and these hydroponics, aeroponic systems. You have disease issues. They're not able to get the microbes the way they would in the soil, which is, of course, where they have evolved to develop. That's where they grow. That's how they have developed over all these millions of years.
Mm-hmm.
When they grow in soils, to use your word now, Everest,
Yeah.
they have agency. They have agency, right? They're able to do what they need to do. They're able to attract those microbes, detect, put the nutrients out there will attract the right ones depending on what they need and bring them back in and internalize them and so forth. They're able to manage those microbes, but when you put them in a hydroponic system, they're not. And the human suddenly now has to guess, and we're not as good as. Sorry, we have big brains, so we're not as good as a plant at figuring out what that plant needs. So that's the issue with that. But people are going to do that. It turns out that it's not as economical as growing plants in soil.
Aha.
Because of all the input, all the light you have to put in, and all the putting a lot of nutrients to manage them, and you get disease. You got disease coming in because they're not stress hardy, because they're not interacting with microbes internally, so they're not oxidatively stress hardy. So now you get a lot of disease. Now you're putting in more chemistry, right? You're putting in fungicides in the system. That's the system. And the food is not as good
Hmm.
as when it's grown in good healthy soils.
Yeah. What do you think, Max? Like, how would you sell somebody who wants to start an aeroponic indoor grow for their
Yeah.
own personal consumption? How would you change their mind?
Yeah. Uh, I tackle this from I mean, I'll tackle it from a less scientific point of view, in the sense that for me it's very simple when it comes to working with living systems. What I always try to do is I try to learn the best way that nature does it, so that I can inspire that. I don't really agree with the philosophy that we can do it better than nature, which is seems to be like such a huge part of this industry. And so yeah, I mean it's this same thing that James was saying, but basically input to output. I always end up on this when we discussed in Everest, you and I, the fact that sustainability, and it's the bottom line for me. I am always going to go to the foundation of input to output. So diverse systems, and where inputs get recycled. I mean, none of that happens in a hydroponic system. I have worked with huge greenhouse systems. I have worked with aquaponic systems. I have had the pleasure and the budget to go deep into these systems. And I know, in my opinion, it's all nothing compares to doing it closer to nature. If you want to close a loop, then you're going to have to learn from nature. Humans are just never going to be able, our systems are never going to be able to live up to it. All those aquaponic systems, hydroponics, all these things. I mean, the amount of input that you need just to grow a plant, in my opinion, it's never worth it. It comes down to entropy within a system at the end of the day.
Hmm. Yeah, and I guess ultimately too, you're pretty limited as to what species you can grow in an aeroponic system, anyways. So it's kind of a moot point in some ways. If you want blueberries, you're not going to have a choice other than blueberries outside, right?
Yeah. Yeah, and just the amount of resource that goes into even just these structures and these,
Mhm.
and it goes on and on and on and on. And
Yeah, true.
A
So,
lot of those, a
yeah,
lot of those businesses are going out of business, by the way.
I've
yeah,
heard.
yeah, they
They
are.
can't make profit. They can't make profit.
Yeah, yeah,
Yep.
yeah. James, you mentioned something. Let's see. You mentioned that people will, okay, so the plants need well, you said they don't need microbes, okay, but then you also said that the microbes, it's hard to guess what microbes the plants need. And that leads me to a interesting question. I think it's a hot topic of AI these days. I was curious, your experience, have you guys been have universities been using AI, for example, researching as a method to research and or identify organisms to expedite research? Is there any application that you see there, or do you think we should not necessarily rely on that?
Well, I've used AI to help um, correct writing for people, because it's easy to get AI to fix if some writing looks pretty crappy. I might feed it into an AI program to
Right.
see if the AI can do a better job on that paragraph, right?
Yeah.
Right,
Which is
right.
kind of fun to do, right? Fun to do,
Yes.
but you have to be careful there, because AI does stupid stuff
Yes.
plenty of times. So you have to make sure that it really is correct. You got to double check the AI. There's the issue. You got to check the AI, double check it. That's pretty good, depending on the program. Now in terms of creative activity, no,
Right.
AI is useless for that. It
Oh,
is
wow.
not going to be able to. It is not going to be able to replace humans in research. You might be able to number crunch. You might be able to AI might be able to bring out some synthesize some stuff, but it's not going to synthesize anything new. It's
Mhm.
only going to be stuff that's already out
Right.
there.
So
You're not gonna get, yeah, and that's what science, the real important contributions in science are new, not the rote stuff, not
Right.
AI. I mean, not just finding what people have found already. And if something is wrong, you see, here's the other problem. If something out there is wrong, AI doesn't know that. AI picks it up and puts it in there. And I mean, you've heard the systems where AI is you ask AI uh, show what a beautiful person looks like, and they end up having one race. One
Yeah.
race is beauty, right? One race is beauty. This is this is crazy. AI, and you look at images at AI. I don't know if you've noticed on YouTube and stuff. You can see these AI generated images, and they're always screwed up. They have
Artifact.
something. There's an artifact. An elephant has 5 legs, or the smoke is coming out of a chimney. The snow is going one direction. The smoke is going the other. AI that's not smart enough to really look at the whole picture. And I know people think it's the future, and if sure it'll be there.
Solve climate change, right?
I don't think so.
Consistent.
I don't think it's going to change human behavior. No, no.
Yeah, because I had imagined it. I had been imagining it as the application being like a morphological recognition, and so where you could actually use it to quickly analyze microscope morphology and maybe even catch something that the human eye misses. But to your point, perhaps it would cause more error than it's worth it.
I think it would be difficult to train an AI with the how the capacity that humans have to pick things up, and certainly for any kind of insight or any kind of intuition, right, about something. AI will never have that. It'll never have that human. I mean, it is basically, computers,
Right.
you know, to, try
It
to.
won't have inspiration.
It's not going to have inspiration now.
Great way to put it. Yeah,
Yeah, that's a good way.
started
Good
this.
way. Good way. Good way. Yeah.
Yeah, it is truly a lot of, yeah, like you're saying, a lot of new discovery starts with an inspiration. So
And only people get that. Only humans. I mean, we can screw ourselves up, but we also are really, all of us are really smart. We're really smart, and we can use that smarts in ways that I think AI never can. We could use AI to number crunch and to process a lot of stuff, but even that takes time. I mean, I've heard training AI is taking so much energy that we're already using, certain percentage of the energy we have to train AI programming. It's already costing a lot just to train them. And then running that constantly, are we even going to be able to replicate it over time to use AI to the extent throughout our society? And I don't know. We just have to see. But I do know for sure,
yeah,
AI cannot replace human creativity.
beautiful, beautiful. Um, so I want to actually now back up one thing. Have you heard of fourth phase water, Gerald Pollack's research on fourth phase water? How much of that is related to these endophytes and the cycles and the plant metabolism and such?
It's hard to say, but they certainly may be related. I mean, things like, for example, movement of these bacteria in phloem or in xylem, moving in the xylem, could in fact be related to how the water and the microbe particles interact. Because what you have basically in the even in the plant cells, I talk about that movement, right? This is all happening underneath the cell walls, and so
Driving
you
that.
have you have substances. You have like the microbe themselves interacting with water, causing water to essentially be constrained, right? And then you have cell walls also interacting with water, because these biogels, that's what they do. They interact with water. And you got biogels on the microbe, and you got biogels on the plant. So they both interacting with water. And what that causes is a conflict, those interactions. And the most thermodynamically stable way for that conflict to break is for movement to happen.
Mm.
And
Wow.
so movement of the microbe could be happening because of that fourth phase water, right?
Fascinating.
Yeah.
Thank you. Are there any unanswered questions that for you that you hope more research can be put towards?
Well, the big things that we're looking at, three things I'll mention. One is we want to look at the evolution. We want to go way back in time, right? Going back, we're looking at early evolving plants, looking at those plants, like things like mosses and moss relatives, very primitive plants, to see how they do it, see what they're doing, to get the idea, where it started. It started way back then, it appears, even back to algae, back to algae. And to show that we have this microbial farming happening all the way back from the earliest plants, that emphasizes the importance of this process in plants. Okay, so that's important.
Beautiful. Yeah.
And another thing is we want to show how plants are doing it. So look at the different kinds of cells and tissues, and I alluded to this already, inside the plant, how they're getting nitrogen from those plants. And finally, the third thing is we want to be able to create crops and fortify crops with microbes so that you don't actually have to have all the chemistry. We can reclaim, rebuild crops so they're close to nature. They have the right microbes in them so that you could grow them without all the chemistry. Okay, with minimal chemistry. Okay, and as a practical matter, I'm collaborating. My lab is collaborating with Walter Goldstein at the Mondawmin Institute in Wisconsin, who's breeding some landrace corn, trying to make hybrids of this landrace corn, like stuff from Mexico. We're talking about, getting, they fix nitrogen and so taking those plants, showing how they fix nitrogen, show inside what's happening, and then helping Walter breed these new varieties so that people can then grow these microbial rich corn types. So
Right.
so those are the kind of three areas that I think are significant that we're working in.
That's very significant. Thank you for that.
You're welcome.
Yeah. Well, we've been going for quite some time now. I guess we're at like an hour and a half, right? So I don't know. I guess we could start wrapping it up, guys. But
You got time for one practical
Yeah.
question
go ahead, please do.
for any listener, grower, gardener, farmer, from what would your advice be for anybody who's really trying to improve their biology in their growing system, as far as I know my go-tos are, using succession and different methods I've developed over the years to speed it up in combination with organic material that will turn into organic matter. And I focus a lot on the microbial sprays is what I spend most of my time studying within my systems, is how to brew for the different populations of microbes and get them on there. What would you say to somebody, what would be the go-to? Would it be a compost tea? Would it be a compost itself? What would be the
Extract.
go-to that you would advise somebody?
That's a
All of
great
those. Yeah, those are good questions. All of those. And those questions come up a lot. It's all of those things. All of those things work. Even things like with Korean natural farming, right? They'll sometimes get endophytes out of the plants by making a kind of an extract where they use bait. They'll put starch in with plant material and get the microbes out and then take that and put those on plants. So it's kind of those kind of
The
systems,
ferments.
that ferments work really well. Yeah, compost teas and washes, building up soil using compost, using, then I always say, and this is the first point, but that is follow the principles of regenerative agriculture, because it's all right there. It's all right there. That's more for, bigger agriculture, not for gardening, but certainly with gardening, we're talking about compost and it's ideal not to have bare soil, things like that. So some of those principles still apply for gardens.
Yeah.
Mixed crops. I think having different species together in the same beds is a good idea, actually, kind of like
Mhm.
indigenous Three Sisters approach, right? Sort of
Yeah.
thing. That's a good idea because they share microbiology. So yeah,
So,
Yeah,
one
I
of the
think
big issues.
diversity is always a plus.
Yeah.
yeah, yep.
One of the big issues with conventional agriculture and switching to regenerative ag, right, is like it's hard to get away from the Roundup when it comes to monocropping or and if we want to be practical, at least my opinion would be that it's going to be a long time until this kind of awareness knowledge leads to a halt of the monocrop system, even if we want to say like, okay, let's intercrop and do cover crops. Well, you still have to terminate the cover crop with Roundup. I'm curious your thoughts on Roundup and where that fits in. Do we need to completely stop using it? Is it that big of an issue, or is it okay as long as it doesn't end up in the final product?
Honestly, I I know this. What you said is correct, and that is I've heard plenty of times that for a lot of growers, there's no solution except Roundup. There's no solution. So, when you have a case like that where there's no solution, but at the same time, you have lots of evidence that uh, it's not good.
Mm-hmm.
I mean, there's downsides to it, and among those downsides are you getting Roundup everywhere, it's not good for people either. So there's no solution yet. What is this long term? Is the development. If people need other kinds of herbicides, then we have to develop other kinds of herbicides that are more consistent, more do less damage to the soil microbes and to the plant microbes. Roundup does a lot of damage. In fact, it was one of the first patents was as an antibiotic, right, to kill microbes. Okay, so but it also will bind microbes, and it's possible that, well, it binds iron too. It binds manganese and iron and other kinds of divalent metals, and binding those makes it so the microbes can't get them. And also it will bind iron, and iron is actually critical for the plant to produce reactive oxygen to get nutrients out of the microbes. So essentially, Roundup paralyzes the rhizophagy cycle because the plant now cannot make superoxide to get nutrients out of those microbes. So but still, it is a chemical that people are using, and they say they need it, and there is no replacement. So, I mean, for me at this point, my conclusion is we really have to develop some new
Alternative.
alternatives. Right?
And change the system, because they're depending on it in a broken system, and that we know from everything biologically it's broken, right? So would they need it if they went ahead and shifted there's the whole soil food web beneath their feet? They might not need it anymore.
Yeah. Yeah. Part of it's for like terminating crops, though, like to terminate a cover crop, for
Cover
example,
crop. Yeah.
in a no-till
Yeah.
system where because like I was just in Maryland and they don't till. There is a no-till state. I'd never heard of that. But yeah, they're not allowed to till their soil, so they use the Roundup to terminate the crop and
It's a
even
horrible
cover
idea.
crops, but they can then drill in the next the corn, right? Like terminate the cover crop, drill in the corn. And so, yeah, I'm super interested in the solution to that. I hope that, is it roller crimping? Could we terminate with just roller crimping? Could we find some sort of an enzyme that would, that's maybe microbially derived, that could senesce plants so that you could terminate early? I don't know. I'm curious about that.
It'd be a
Well,
matter of finding the right crop to grow it with.
Yeah.
and I think we need some, as we said before, new herbicides. We need some new herbicides,
Yeah. Yeah. Mm,
new
Okay.
strategies even. There's other ways to kill plants.
Yeah.
To terminate a plant, there's other ways to do it. And you can actually do it microbially.
Mhm.
And I'm not going to go into any details, but if you study the interactions between microbes and plants, you could pick up clues about how you could actually terminate that plant using
Wow.
microbiology.
Okay.
That sounds like
go.
something one of these big companies will take into their own hands and can create a whole lot of trouble. Sounds like.
Well, if it's not harmful to people,
Yep.
if it's food grade stuff in it, right? If it's edible, not deleterious to the health, if it stimulates microbes to do a certain thing to the plant, and then when that substance is gone, then all you have left are the microbes. Then there's no problem. There's no problem to human health, no problem to the environment. In fact, you enrich the environment.
Beautiful.
You can
That's what we need to look into. That's the solution.
Right.
it's possible to do
Right.
that. It's possible to do that.
Epic.
That
Epic.
sounds like
Thank
the
you for
future.
saying that. That's huge.
I think it could be the future, but
Yes.
we have to wait till the future gets here.
Right.
Yeah.
Till the future is in the present, and then we'll look and see, right? When that happens, we'll look and see if it really happens.
We're counting on you for it.
I know, right? Let us know how we can help support.
Yeah, yeah.
I want to be on the front line of that.
Well,
thing.
fingers crossed.
Yeah.
Fingers crossed.
Okay. Awesome.
Wow. Well, thank you so much.
Thank you.
You
Really
guys
appreciate your
so
time.
welcome. I enjoyed it.
This was
Very valuable to us, our time with you. So thank you.
Yeah. And yeah, hopefully we'll stay in touch and
Okay.
continue to follow your work and continue to spread the awareness of the intricate interrelationship between microbes, plants, animals, humans, the soil. We need to build soil. We need to save the soil. Maybe it's not a huge existential threat right now, but it's the solution to a lot of what
To our problems, it really is.
Okay.
Yeah. Yeah. Okay, so you guys have a great afternoon.
Thank you so much.
Thank you so much. Have a good